last speaker of the session is Mr. Ayman Abuelela, and he will talk about ENCODE ChIP-Seq analysis.
First, I'd like to thank the organization committee for giving me the chance to present some of my work. Actually, the main motive of this project is the fact that metastasis is the major mortality cause of cancer patients. And in this brief talk, I will try to build the association or explain the association between Sialyl Lewis X and A and metastasis, and our trials to study the regulation of Sialyl Lewis X and A expression. So Sialyl Lewis X and A are tetrasaccharides born on mucin proteins and also glycolipids of epithelial cancers. And actually they are the binding motif of some adhesion molecules called selectin family, and we are interested in the endothelial selectins E-selectin and P-selectin. And the difference between Sialyl Lewis X and Sialyl Lewis A is mainly the substitution on N-acetyl glucosamine on carbon 3 and carbon 4. So fucose and galactose are reversed on Sialyl Lewis X, on Sialyl Lewis A in comparison to Sialyl Lewis X. What is the role of Sialyl Lewis X in metastasis? Actually, in 2008, Saxtain hypothesized the hematopoietic mimicry model, in which he stated that circulating tumor cells resembles immune cells in the way they extravasate the circulation towards inflammatory sites. So actually, circulating tumor cells expressing glycoproteins that has Sialyl Lewis X will be able to bind endothelial selectins and then undergo subsequent adhesion interactions till extravasation and formation of the secondary mass or metastatic tumor. Not only that, it was proven, but also it has been proven that the first transition programs at the primary site also involve upregulation of Sialyl Lewis X, and then these cells can be more invasive and more motile and undergo intravasation into the circulation. And also in the circulation itself, Sialyl Lewis X helps circulating tumor cell to interact with platelets and leukocytes to form metastatic emboli. And actually this helps metastasis in two ways. The first way, it protects the circulating tumor cells to evade the immune system attacked by the immune cells, and also it can bridge the interaction with endothelial selectins. And all that will help the extravasation of circulating tumor cells to metastasize and form a new tumor at the distant site. And it's believed that Sialyl Lewis X and its interaction with E-selectin is one of the factors that dictate organ-specific metastasis of epithelial cancer. But now we have a problem. Sialyl Lewis X is a carbohydrate, and carbohydrates is not directly encoded by the genome, but they are biosynthesized with a specific group of enzymes called glycosyltransferases. So in order to study the regulation of Sialyl Lewis X expression, we need to study the regulation of glycosyltransferases involved in the biosynthesis of Sialyl Lewis X and Sialyl Lewis A. They are mainly 4 enzymes, and we are interested in 3 major enzymes. The core-2 branching enzyme that puts the N-acetyl glucosamine branch on the protein, and 2 major terminal decorations, the fucose decoration and sialic acid decoration. So we are interested in fucosyltransferases, sialyl transferases, and core-2 forming enzymes. And before that, we studied the expression of glycosyltransferases in 2 breast cancer models, BT20, which is Sialyl Lewis X positive model, and the MCF7, which is Sialyl Lewis X negative model. And actually we found a good disparity in the expression of glycosyltransferases between two models. As you can see here, the relative expression between BT20 and MCF7 shows some upregulation of some sialyl transferases, fucosyltransferases, and COR2 forming enzyme. And now the question, how Sialyl Lewis X is regulated, how the glycosyltransferases are regulated. So our aim was to to study the regulation at the transcriptional level, so to identify the transcription factors upstream of glycosyltransferase genes, and our hypothesis was that the transcription factors with higher frequency of occurrence upstream of glycosyltransferase genes will be most likely controlling this biosynthetic pathway. So for example, if I found that one of the transcription factors is controlling 8 isoforms out of 11, so most likely it's controlling the biosynthetic pathway. And in order to do that, we used the open source ENCODE ChIP-Seq transcription factor data, and we used two inclusion criteria for the transcription factor ChIP-Seq hits. The first is the transcription factor must lie within 1.5 kb window, 1 kb upstream of the transcription start site, and half kb downstream of the transcription start site. And the second criterion is it must lie in DNase 1 hypersensitive region. And we started collecting the ChIP-Seq hits and collecting the scores and we classified the glycosyltransferase genes into 3 classes. The first class is all the glycosyltransferases, or the 3 glycosyltransferases. So transcription factors in this group will be most likely controlling the whole pathway of the biosynthesis. And actually you got a repertoire of around 90 transcription factors. And the second group and the third group are the key decoration of the lactosamine unit, the fucose decoration, and Sialyl Lewis X decoration. And the top hits of the 3 groups including some, they include some interesting hits, like the major hits in the total glycosyltransferases are c-MEC and ELF1 controlling 8 isoforms out of 11, followed by HEY1, FOXA1, FOXA2, MAX, and CEBPB, which controls 7 isoforms out of 11. So maybe those transcription factors have a major role in controlling the biosynthesis of Sialyl Lewis X. And in case of fucosyltransferases, after c-MEC and ELF1, we found that again FOXA1 and FOXA2, CEBP are controlling the expression of or lying upstream of fucosyltransferases genes. And in Sialyl transferases, we got TAF1 and YY1 and USF1. And actually we were interested in the question of what are cellular processes or cellular programs are involved in the or associated with those transcription factors and all our repertoire of identified transcription factors. So we did functional classification of the identified transcription factors, and we found interestingly that most of our transcription factors are involved in the morphogenesis programs and embryogenesis programs. And as you know that tumor cells at the primary site needs morphogenesis program at the first level to change from the stationary phenotype to the motile invasive phenotype. And actually this group includes the famous epithelial mesenchymal transition program involved in supporting metastasis of cancers. And also interestingly, we found transcription factors involved in the differentiation of immune cells, because actually immune cells share the same mechanism of Sialyl Lewis X to extravasate when going to inflammatory sites. So actually this is very interesting. And actually we found some hits involved in proto-oncogene and in tumor progression and metastasis. And interestingly also we found some hits involved in cell stemness. And actually this is outline of the transcription factor involved in three major cell processes: stemness, morphogenesis, and epithelial mesenchymal transition. We believe they are involved in controlling and supporting metastasis. So just to brief, Sialyl Lewis X forming glycosyltransferases lies downstream of a defined group of transcription factors like c-MEC, ELF1, FOXA1, FOXA2, HEY1, MAX. Those are the top hits of our study. And we believe that the identified transcription factors are associated with prometastatic programs like morphogenesis and epithelial mesenchymal transition, also associated with cell stemness. And actually, there is a growing debate now in the community that cancer stem cells are one of the major factors of initiating distant metastasis. And also we found some hits associated with tumor progression and proto-oncogenes. This study, we hope it will permit further experimental investigations, and now actually I'm in the phase of preparing some stable transfectants of our top hits, and actually trying to do some expression experiments to correlate between glycosyltransferase expression and transcription factor expression. And our ultimate hope is that we can identify novel poor prognosis gene signatures associated with metastasis. Finally, I have to acknowledge the ENCODE project consortium and all my lab members and my advisor, Dr. Yasmin Mirzaban, and thank you.
Thank you for a very nice talk.